Hi, my name is Jess Holtz, and I'm going to be talking about fluorescence microscopy, in which photons excite fluorescent molecules in the sample to emit these bright glowing lights. It's used a lot in biology, but I'm using it more for art. In this case, we're looking at plankton from Woods Hole in Cape Cod, Massachusetts, under a triple bandpass fluorescence filter. Triple bandpass basically means seeing three colors at once. In this case, we have this bright green fluorescent copepod. So seeing three colors at once as opposed to one at a time, like is commonly done. In this way, I can record video of these creatures in all three colors. And I'm recording their autofluorescence instead of staining them. You can see a lot of crustaceans and algae. The chitin in these copepods and crustaceans fluoresces blue, and the chlorophyll in the algae fluoresces this bright glowing red. I'm using a Leica DMRB with this triple bandpass filter, as well as a UV long pass. So this is what these fluorescence filters look like. They are placed inside the microscope. There is an excitation filter to pass through a narrow range of wavelengths to excite the fluorescent molecules in the sample. There's a dichroic for keeping that excitation light from getting into your eyes and the detector. There's an emission filter for letting a range of emitted photons from the sample through the detector. Now I'm going to be comparing two filters, a UV long pass and a triple bandpass filter from Chroma. So here's the UV long pass, and here is the triple bandpass. So with the UV long pass, it basically has this UV excitation band from 350 to 400 nanometers, and then it just lets everything through after it. The emission just lets everything through. This is in comparison to the triple bandpass, which has three separate excitation bands, three different colors, violet, blue, and yellow-green. And then there's also three bandpass emission filters. So I've really had fun with this triple bandpass filter. This is some pond water from Hall's Pond in Brookline, Massachusetts. And I can just see these creatures fluorescing as they are alive with the triple bandpass filter, as well as the UV long pass, but it's not quite as colorful. And for the rest of the video, I'm going to be comparing the UV long pass to the triple bandpass just to see the differences that these two filters have in how they fluoresce. So back to the marine water, the ocean water from Cape Cod. Here we have the triple bandpass and the UV long pass. I'm very interested in the differences that these two filters have. So again, you can see the chitin in the copepod fluoresces this blue color. It's a bit more muted in the UV long or in the triple bandpass. But now we have dark field, which is more traditional microscopy, non-fluorescent microscopy. So here we have, in a second, here we have some microplastics in the water, and I've found these in a lot of different waters. It's kind of sad, actually, and they fluoresce a variety of different colors. They're very colorful, and they're like these bright, glowing rods. Some can be very, very bright. Here we have a snail on the left and some algae under the triple bandpass. The algae just gets really, really bright. The chlorophyll in it fluoresces red. Yeah, so just showing you this comparison between these two filters from Chroma. I get these from Chroma filters. Here's a Nauplius, a copepod larva. Now this is a different triple bandpass filter that is not well matched to my excitation light. It's made more for a mercury lamp. So I'm returning that one. Then here's the UV long pass. Here's some copepods and a microplastic. some trichromes on the top left, along with algae. I'm really interested in this yellow color coming from the UV longpass, but that turns red in the triple bandpass. Another copepod larvae. And here we have, I believe it's a flatworm under the triple bandpass. And I'm really interested in the different types of information you can get from these two filters in dark field now. You can see the gut is glowing with something, not sure what, is green or white in the triple bandpass. But yeah, that concludes my video. Thanks for watching.